happy to share with you some of the recent advances which are made in biotechnology. And as per confining to your topic, genetically modified plants for improved tolerance to biotic and abiotic stresses. Let us see uh, what are genetically modified plants. As the name indicates, they are modified just by from their genes or something related to nucleotides, etc. So the plants into which few genes, one or few genes, are transferred from another plant or another microorganism or another any organism using the techniques of recombinant DNA technology uh, are called as genetically modified plants. Now, when you are transferring genes, why should you transfer useless genes? Always you go for the novel genes, which will give certain capacity to that organism, new capacities, that otherwise that capacity was not there to that plant or animal earlier. So, or alternately, you can remove certain harmful genes. If a host plant is having some harmful genes, that genes can be removed using this DNA recombinant DNA techniques. This generally we call it as genetic engineering. Either you can transfer novel genes or you can remove few harmful genes. Either way, that is called as genetic engineering. But only transferring genes from one organism to the other, useless, useful genes from one organism to other organism is called as recombinant DNA technology. So these genetically modified plants, after transfer of genes, they will become a newly potent with new capabilities. Those plants they will have. Maybe they may be a disease-resistant plant. They may be drought-resistant varieties. So like that, some are the other new characters they will acquire. These GM plants or genetically modified plants are also called as genetically modified organism. Organism is the word used for both all living organisms. They may be plants, they may be animals, they may be microorganisms. When you are talking about plants, we generally call it as genetically modified plants. They are also called as genetically engineered plants because genetic engineering techniques we are using and we are modifying them. So they are called as genetically engineered plants. Bioengineered or transgenic plants. So these are the synonyms, other terms used for describing these genetically modified plants. No, modifying genes is nothing new today. It has been continuing since ages. People they wanted to have very good horses with desirable traits, very good cows which can give more milk, or crop plants which can give good yield, disease resistance plants. All these we have been using other than genetic engineering techniques. Earlier, they used to take domestication, selection, and hybridization. Such techniques they used to follow. All of you are very familiar with the classical plant breeding technique. Classical plant breeding technique. So the plant breeding, when two plants, suppose one plant has high yielding capacity but low disease resistance, the other plant has high disease resistance but low yield. People they used to cross hybridize these two plants so that they will get good characters of one plant and the good characters of another plant into a single hybrid plant. So this may be reverse also. You may get bad characters of one plant combined with another bad character of the another plant. We may get a useless hybrid. So you have to sow in the field several generations. You have to take select which are the novel ones, which are the good hybrids that you have to select. So this is very time consuming process. People used to hybridize the plant, wait for a F1 generation, F2 generation, F3 generation. Like that, generally it used to take 7 years to take out a very good hybrid. So this is less precise. We don't know which genes are transferred there, which genes actually are not transferred to that plant. We don't know anything unless and until go to the field, observe the field, find out which character the hybrid has acquired or which characters it has lost. So it is less precise. There are species barriers. All plants you cannot make it hybrid. So when you make hybridization between two plants, that hybrid may not be successful. There may be species barriers. Yield plateauing. Hybrids also they give good yield up to certain generations. After that, that yield will become static. That is called as yield plateauing. Inbuilt biosafety regulatory systems are there. All these they make the classical plant breeding system, even though it is good, it is not that time consuming or it is not that time saving or precise like that. So nowadays, here I think I try to show a small comparison. Traditional plant breeding is that traditional plant breeding combines many genes at once. For example, this is donor plant. This gene I want to transfer. The yellow color ones are other genes. These genes, this gene I want to transfer into these green color ones. When I make hybridization, that desired gene is transferred, but along with that, some undesired, unwanted genes may be transferred into this. There is no precision. Accurately, one gene you cannot transfer. Whereas now, in the era of genetic engineering or recombinant DNA technology, it is now possible that you can add a very single gene very precisely to the DNA strand of the other plant. So here, just like earlier plant, yellow are the other genes, and this one, bluish, is a desired gene. This desired gene can be isolated from this donor plant. Only one gene can be isolated. It can be purified. It can be multiplied. It can be made into thousands of copies. And these thousands of copies will be combined with commercial plant variety. So some of them, they will receive this very accurately, the single gene, desired gene they will receive. So that we call it is a improved commercial plant variety, or simply we call it is genetically modified crop, genetically modified plants. So that precise is the modern genetic engineering, biotechnology, or recombinant DNA technology. What is real DNA recombinant DNA technology means? The technique of breaking DNA molecules at desired places to isolate a specific DNA segment. That specific DNA segment will contain a desired gene, and then purify it, multiply it, and insert it into another DNA molecule at the desired position is called recombinant DNA technology. The product thus produced is called as recombinant DNA. The whole DNA means the foreign DNA plus the native DNA combination is called recombinant DNA. Native DNA means the commercial variety DNA, and foreign DNA means the desired gene which you have isolated from a donor species. So the plant into which a desired gene is transferred, employing the techniques of recombinant DNA, is called a transgenic plant. What types of genes can be transferred? Practically any type of gene. You know, most of the characters are controlled by single genes. Qualitative traits, qualitative characters are controlled by single genes. So the qualitative genes, if you want to transfer, it is easier. Qualitative genes like increased protein content, improved seed storage, altered fruit ripening, disease resistance, insect resistance genes, herbicidal resistance, stress tolerance, all are controlled by single genes. Whereas yield, yield is a quantitative metric, quantitative trait controlled by several genes. So several genes, isolating them, combining them, that may be a bit difficult as on today, but most of the work has been done on transferring single genes. Is an imaginary. What do genetic engineers do? How do they transfer single genes? In the picture we have seen, suppose one red pigment gene is to be transferred. If the red pigment is desirable, if it is good, if it gives you some commercial value, you can transfer it. Suppose that gene, if you can transfer to bacteria, the bacteria may they become red. It can be transferred to banana, flower leaf, flower plant, frog, practically anywhere you can transfer. Once you transfer, what will happen? That gene will go into that organism. There it will be expressed. Once it is expressed, we feel that our gene transfer technology is a success. So genetic engineers carry out direct manipulation of an organism's genes using techniques of biotechnology. And to be precise, they are called as recombinant DNA technology or techniques of genetic engineering. So what they do, they identify particular useful gene. Identify how we identify that gene that we study in isolation of gene, like that. I think in a short summary, I will explain that also. So they identify particular useful gene, isolate it, clone it, and transfer it into a construct. A DNA construct means a DNA molecule. Into that they will send. This construct, matlab DNA molecule of the commercial variety in which a new insert has been taken from the donor plant together is used to multiply further. So recombinant DNA technology has got three major steps. One, isolation of gene of interest. So this is usually taken from donor. Cloning of isolated genes, we call gene cloning. Transfer of cloned genes into the host. These are the three important steps involved in recombinant DNA technology. Now, in our topic, genetically modified plants for biotic and abiotic stresses. What is biotic stress? Biotic stress means, for example, cotton plants are there. Cotton plants are infected by several insects. Some larvae they will eat the birds, the pods, like that. So it will get so much stress. How to fight with these insects? How to protect myself? So the plant has several resistance mechanisms here. So if you can transfer certain genes which can give disease resistance, pest resistance, so that will take care of the biotic stresses. What are abiotic stresses? Abiotic stresses are non-living stresses. Like this, for example, they are drought resistant. In some area, water is not there. Water is not there, and the plants that try to survive with little water. So they face a stress, water stress. Similarly, salt stress. Like that, whatever the general non-living stresses are there, they are called as abiotic stresses. Here in this example, I am giving a BT gene transfer. How isolation of gene of interest, that is a uh, uh, pest resistant gene, how it can be transferred from one organism to plants, how they can be cloned, how they can be transferred into the host. So, first step is isolation of gene of interest. What example we are taking? Isolation of BT genes. What is BT? B stands for Bacillus, genus name. T stands for Turin genesis. So, BT gene is a gene from Bacillus thuringiensis. So I try to give a rough diagram. I think that these are the bacteria. All the bacteria are crushed Bacillus bacteria, this Bacillus thuringiensis, isolate their DNA. So you will get all DNA, total DNA, genomic DNA. You digest with restriction endonucleases. I think in earlier classes, even in MSc and others, we have studied what are restriction endonucleases, what are their specific characters, how they are used, for what purpose they are used, like that. Once EcoR1 is one of the such restriction endonuclease, they cut the DNA into several pieces at particular restriction cleavage sites. So the result is the whole genomic DNA is broken into pieces. The whole DNA is separated. Here, separate the fragments by agar-gel electrophorese. When you separate, make two slabs, agar-gel electrophorese slabs. One gel slab you will use for Southern blotting technique for identifying. Suppose out of these two, this is the first one and this is the second one. In the second one, using Southern blotting, you will identify which gene has got this BT, which DNA fragment has got this BT gene. So this BT gene is cut and separate from the corresponding band from the second gel. So in the first gel, if you identify here, the corresponding one is cut and isolated. So it contains the BT gene. This single gene, the single gene is now taken for gene cloning. It is multiplied several times. Nowadays, people are using polymerase chain reaction for multiplying these genes, gene cloning. I think without wasting much of your time, I will skip to our main lecture. So earlier they used to use cloning vectors. Now cloning vectors also they can be used, or in place of it, if you use others, that is more economical. So these cloning vectors have several types: plasmid vectors, lambda phase vectors, plasmid vectors, and fourth one is artificial chromosome vectors. Each vector has different capacities, load-bearing capacities. Plasmids, they can bear the load of 5 to 10 kilo base nucleotides. Lambda phases, they can bear 23 kilobase weight of DNA insert, whereas large insert, biggest load is 1000 kV, yeah, 1 MB. That nucleotide can be born by the yeast artificial chromosomes. So like that, we have different cloning vectors. They have different load-bearing capacities. Here, a small picture, it indicates somewhere this is the plasmid cloning vector. In this cloning vector, Ti plasmid has taken. It is digested with E carbon. In this place, cloned Bt gene is mixed, ligated with T4 gene. So Bt gene is here, and this is called as recombinant plasmid or chimeric plasmid. This plasmid is inserted into agrobacterium. In agrobacterium, bacteria divide 1 into 2, 2 into 4, like that. Many bacteria are given. And at a certain stage, all the bacteria are killed using kanamycin. With kanamycin, all the bacteria will die. And this plasmid containing the Bt gene is used for transforming the plants. Here, all these bacteria carrying cloned genes and leaf discs are taken. Suppose this is a cotton plant. From cotton plant, cotton leaf disc is taken. In tissue culture, this is infected with bacteria. They culture it. In tissue culture, shoot initiation, root initiation will be there. At certain place, with kanamycin, all the bacteria will be killed. The plant will grow. The plant gets incorporated those Bt genes, and this is called as transgenic plant or genetically modified plant. This plant will acquire a new gene. Which new gene it has acquired? Bt gene. So Bt gene, Bt Bacillus thuringiensis gene, actually it is a poison to most of the insects. So this cotton becomes Bt cotton. It produces a toxin. The toxin is for Leptopteran insects. It is a deadly poison, not for you and me or any mammals. Okay, it acquires. So genetically modified plants for improved tolerance to biotic stresses. This is our actual lesson. Let us see few examples. As I told you, insect resistance is provided by a Bt gene. Mostly nowadays, whatever insect resistance plants are produced, all this earlier they used to transfer Bt gene. Now, in its place, another genes have come, cry genes, crystal genes. So that crystal genes they are transferring. So Bt insect resistant crops. The use of Bt to control insect pests is not new. Insecticides contain Bt and its toxins. In Bt toxins, there are three types. Dipel is a protein. Turicide is another protein. Vectobac is another protein. All these proteins are sold for several years. All these three protein toxins, they are present in Bacillus thuringiensis. Now, what is new in Bt crops is that a modified version of the bacterial cry gene, crystal coat gene, has been incorporated into the plant's own DNA so that the plant's cellular machinery produces the delta endotoxin as a part of the plant's normal development. This has led to the production of insect-resistant Bt transformed lice. So these two earlier experiments, they used to transfer Bt gene. Nowadays, commercial crops and all, instead of using that Bt gene, they are using these chryzines. There are 9 chryzines available, different chryzines. We will come to that. So here American bollworm, which is also called as cotton bollworm, Helicoverpa armigera, causes so much damage in commercial croplands. So this first one is the insect. Actually, this insect lays eggs in the birds. So the eggs they come transform into larvae. All these larvae they attack the pods, the balls, buds, etc. So here bird, ball, and everything. So larvae feed on buds and green balls. Punctures are made. These punctures are larger in size as compared to spotted bollworm. One such large bore you can see here. They feed on ovary. Half part of the body is inside the fruiting body, and half part of the body comes out here and here. You can see. They, due to that, all these spots they flare up, they become dark, and they become useless, and it causes heavy damage to the cotton plants. So the plants are under severe biotic stresses. Now, what is the stage, economic threshold level? At what stage farmers they spray? This one is at least one egg or at least one larvae for a plant should be seen, or 5% of the fruiting body damage is there. Then people they spray these uh, pesticides. This Helicoverpa armigera, yeah, cotton bollworm, feeds not only tobacco but as many as 181 host plants, including 69 crop species. It attacks and causes losses worth 540 million US dollars annually in India. So that much loss. See the how much loss it is causing in India alone. So BT cotton, after the transfer of BT genes or crystal genes, the cotton is produced. This cotton, what happens in the buds when the insect eats the ovary tissue, leaf tissue, or any of the cotton plant tissue, this what we call the venom proteins, poisonous proteins, they go into the gut of the insect, and that insect will die with severe stomach ache, crimpling of its gut, and no chances of survival. So this larvae, they die at very early stage, so that plant slowly regains and gives very good yield. So here some of the pictures taken from the internet, how the BT cotton has changed the economy of the whole scenery of this. So what are the BT cotton effects? BT cotton in United States, yield effect was 0 to 15%. Either effect was not there, or if it is there, it is up to 15%. In China, the yield effect was 10%. Whereas in India, the yield effect was 60 to 80%. Why this much effect? 60 to 80% effect. That means see how much damage, how much infestation the plant, this insect is causing. Thus, that much damage was there. Now, after introducing this BT cotton, farmers farmers they are getting very good yields. What are the environmental implications? Not only this, due to this, the environmental implication also we call it as contribution to environmentally safer agriculture in the world. So after introduction of insect resistant cotton in India and China, yield improved only 6%. Yeah, but yield in general, maybe in India it is 80%, 60% like that. But in most of the third world countries, on an average, it was 6%. The use of pesticide has decreased to 80%. See the vast difference. 80% decrease in the use of pesticide. How much pesticide pollution was decreased? Farmer production costs lowered 82%. So in crores, if you see per hectare, this much, uh, 5,640 is there. So like that, it has saved a lot of our efforts, insecticide use, pesticide use, like that. So these are the very good environmental implications. Once upon a time, most of the BT cotton plants, BT cotton, BT brinjal, they were not accepted by the people. But now, after looking into the BT cotton has been widely accepted by the farming community in the country, which has increased the productivity, reduced the expenses on crop production, and changed the economic status of the farmers. After BT cotton, corn actually it was the first one, then soybean, rice, wheat. Now that genetically modified uh, genetic engineering is entering the world of vegetable crops also. So in that, the first was the BT brinjal. So BT brinjal also, it is called Bacillus thuringiensis brinjal. It is popularly known as BT brinjal. It's a genetically modified strain created by India's number one seeds company, Mahiko, in collaboration with malnutritional, uh, American multinational Monsanto company. It claims to improve yields and help the agriculture sectors. So, similarly, ring spot virus of uh, of papaya in Hawaii in Hawaii Islands. Once in 1995, papaya ring spot virus had devastated the Hawaiian papaya industry. Virus resistance was introduced via recombinant DNA techniques, which has re-established the industry in Hawaiian Islands. Similarly, yellow bean mosaic virus, a resistant strain, was introduced in Bhende. All these are few examples how biotic stress can be tackled by using recombinant DNA technology. So these are the genetically modified plants. Similarly, it is a transgenic tomato, tomato in which some new genes are transferred. The genes which were which confer or which provide resistant to leaf curl virus. So the left-hand diagram is leaf curl virus. So after getting resistance, the crop grows well. It gives a better yield. Like that, many, as we discussed earlier, the genes used CRY1AA, CRY1AB, these are the 7 genes here. All these 7 genes are transferred into the croplands instead of BT genes into 9 crops they have done. This was the old data. The data must have been changed also. So many other plants must have come here. So second part, genetically modified plants for improved uh, tolerance to abiotic stresses. Earlier we have seen about biotic stress. Let us see improved tolerance to abiotic stress. Some examples, let us see. So this crops with resistant to abiotic stresses is also means tolerance plants which have tolerance to herbicide and drought conditions. So weed control is one of the farmers' greatest challenges in producing crops. Most farmers use uh, selective chemical herbicides, excessive use of herbicides, pesticides, all these, they are unnecessarily causing pesticide pollution or herbicide pollutions, and it is becoming an alarming situation nowadays. So, what are the uses of, advantages of using herbicide resistant, genetically modified crops, plants? So, herbicide resistant crops or tolerant crops contain genes that enable them to degrade the active ingredient in herbicides and rendering them harmless. This is called as detoxification. Field you spray with that herbicide, all other plants will die except the plants which have received herbicide tolerant genes. Those that will degrade the herbicide, no effect on them, so they thrive better. And by this, farmers can easily control weeds during the growing season and have more flexibility in choosing times for spraying these herbicides. So, engineering for herbicide resistance, genetic engineering techniques. There are two major approaches. One approach is transfer of this herbicide resistant uh, transfer of a gene which directly do not provide resistance, but it provides an enzyme, it produces an enzyme. That enzyme product detoxifies the herbicide. Okay, directly herbicide resistant genes you are not transferring. You are transferring a precursor gene. That gene will produce a herbicide, uh, produce a enzyme. That enzyme detoxifies the herbicide. Another approach is directly transfer herbicide resistant genes into the croplets. You can directly transfer. So some herbicide resistant genes people have identified, isolated, they are available in the market now. So let us discuss something about these two techniques. Genes conferring herbicide resistance in plants. These are the two genes. The first gene will not directly control that, but it will produce a gene. The gene is CP4 EPSP synthase. Okay, it's also called as 5-enolo pyruvyl shikimate 3-phosphate synthase gene. So that gene will produce this enzyme. This enzyme will degrade that one, the herbicide. Whereas the second is a bar gene. Bar gene, full name is bialaphos. Bialaphos is a herbicide, a resistant gene. So here, the most widely used herbicide, Roundup, all of you must be knowing the Roundup. Roundup is the most widely used herbicide. It contains glyphosate. The glyphosate inhibits synthesis of an enzyme called 5-enolo pyruvyl shikimate 3-phosphate, which is in short called as EPSP synthase, the functionality of which is absolutely required for the survival of the plants. So if you stop this, that weed will die, that plant will die. That is how this, the first CP4 EPSP synthase acts. So based on this principle, Roundup Ready cotton has been produced. Like that Roundup Ready soybean, in general we call Roundup Ready plants. They carry the gene coding for glyphosate insensitive form of enzyme, which is obtained from this gene is obtained from Agrobacterium species strain CP4. Once the gene is incorporated into the plant genome, this produces a gene product which is an enzyme. The name of the enzyme is CP4 EPSP synthase. It gives resistance to glyphosate. Means the plants which contain this, which produces this enzyme, you spray glyphosate, it will not die. Other plants will die. Other unwanted plants, they will die. Like that. Similarly, biolafos is used in agriculture. It's another herbicide, which is a non-selective herbicide. Non-selective means it's a general broad spectrum herbicide for all plants. It can be used. If this if bar from this a gene is removed, this gene is called bar gene. So a gene which confers biolafos resistance is called as bar gene. It has been cloned from Streptomyces hygroscopicus. That gene they have removed. So again, I have mentioned the same thing here. So I will skip this. Now see this here. Here, transfer of gene which can detoxify the herbicide. So see all these plants, these two rows, this is the Roundup Ready cotton, and other on both the sides they have used control in which this gene has not been transferred. Herbicide resistant gene has not been transferred. So the gene is producing an enzyme, and that enzyme, when you spray Roundup, so that Roundup is broken down, degraded into harmless products, and these plants they survive. So this is a Roundup Ready cotton. Here, Roundup Ready cotton plants carry the gene coding for a glyphosate insensitive form of enzyme. So this enzyme is obtained from Agrobacterium species strain CP4 that produces another enzyme CP4 EPSP enzyme. This enzyme confers or gives crop resistance to glyphosate. So whenever you spray glyphosate, it will break down the glyphosate and protect the plant. So this is how the first technique is used. This is called as Roundup Ready cotton. Next one. Similarly, here bar gene, which is taken from earlier I told you from this that genes are directly transferred into the croplands. So bar gene, which gives confers resistance to biolafos. Bar gene, which is resistant to the herbicide biolafos, has been introduced into several plants that allowed them to grow when exposed to the weed killer. So here, in large thousands of hectares, the plants are cultivated, and through helicopters, this biolafos is sprayed. Biolafos is sprayed, so in the entire field, all weeds they die, all weeds. But only the plants in which the bar gene has transferred, they remain unaffected. They grow luxuriously. So this is the second alternate method of transferring these herbicide genes into croplands. The second one. So like that, in these two ways, they are transferring the techniques. So here in both the cases, in both the Roundup Ready cotton and this, we are conferring herbicide resistance weedicide resistance. So the plants, weeds are, weeds honestly they interfere with the nutritional status of the plants. They they will not allow the other plants to grow. So farmers they are fed up with these weeds and how to grow croplands without the fear of, without the stress of these weeds is this is the one of the way. Drought tolerance and disease resistance are another two methods used here, whereas disease resistance comes under biotic stress. So here we need not mention that. Whereas drought tolerance I will take. Here in the second diagram, drought susceptible and drought tolerant palmellate photo is shown. When it is exposed to drought, some of the plants they dry without food they cannot grow. So the plants into which the drought tolerant genes are transferred, they thrive well. So the full summary of here. So they got 5 to 6 genes they have identified for drought tolerance in most of the plants where drought conditions are prevailing, like Rajasthan, Gujarat, other parts. So this drought resistant, yeah, drought tolerant genes are transferred into that. Some of the dominant genetically modified crops in the world are either insect resistant or herbicide tolerance, confining to our topic, cotton, potato, corn, and tomato are the insect resistant crops. Even we have seen even brinjal is insect resistant. Somewhat tomato has already is given here. So like that, many more crops has come. So this lecture may not be up to that date. This is prepared earlier. Herbicide tolerance crops: corn, canola, cotton, soybean, flax, linseed, rice plants, wheat plants. So a herbicide tolerance was provided here. Okay, plants. So like that, many more plants they are using. Here, 16 field crops, alfalfa, barley, canola, cassava, 14 vegetables, broccoli, cabbage, etc., potato, pepper, tomato, 16 fruit plants, and 11 other crops are they have genetically modified plants. Either they harbor some of these disease resistance, insect resistance, or herbicide resistance genes. Other than this, they are using this recombinant DNA technology for other purposes also. For example, nutritional enhancement. If you want to improve the quality of the crop, so this genetic engineering technology. I think golden rice. Golden rice is one such crop. This rice generally vitamin A, so rice eaters, they get a less amount of vitamin A. So people they thought of why not we supplement the rice with vitamin A, carotene, something like that. Carotene gene has been transferred. Carotene is the precursor for this vitamin A. So golden rice has come. Like that, so many plants, they have supplemented the food with iron supplement, maybe B complex certain supplements. Like that they have done. They are also using such methods. So what are the potential benefits of quality improvement of crops through recombinant DNA technology? Reduced use of conventional pesticides. We have seen that point. reduced use of other agrochemicals, fungal and bacterial resistant genes they have transferred, and improved climatic stress, yeah, abiotic stress tolerance, drought tolerance, salinity tolerance, unfavorable temperature tolerance. So, like that, such plants were also uh, produced using genetically modified genetic engineering techniques. Better product quality, higher nutritional value with longer storage. In longer storage, the tomato is one example. Tomato, if the bumper crop has come, it will be perished very easily. You cannot store them for a long time. Now that shelf life has been increased to 3 months by transferring certain genes in tomato. So that was uh, flavor sour tomato that has come to the market also. That is one example here for longer storage. So this is also again summary of what we have. In soybean, herbicide tolerance, modified seed protein storage genes have been transferred. In rice, keratin synthesis genes, insect resistance, modified seed protein storage genes have been transferred. In cotton, herbicide tolerance, insect resistance genes are transferred. Potato is an example where herbicide resistance was successful, virus resistance was successful, and increase in quantity of starch was there. And tomato, virus resistance, herbicide tolerance, modified ripening, and then shelf life increase, insect resistance. These characters were transferred. So gene transfer means gene gives one trait, one character that it acquires a new character. Like that in corn, maize, herbicide tolerance, insect resistance, virus resistance, like that they were transferred. These were the some of the success stories where uh, this biotechnology or genetic engineering techniques were used. So, what are the benefits of this biotechnology or genetic engineering techniques? Means you may get increase the food quantity, you will get better quality of food, and you will achieve better environment where it decreases pesticide pollution, other pollutions, and greenery will be increasing. So all these are the benefits which we can derive, we can reap from using these technologies. Thank you.